Hello friends, today we are going to study about biological oxidation and reduction part 2. Before we learn further about oxidation and reduction in the biological system, let's get a quick review what we learned in the last video. So what we learned about oxidation and reduction is oxidation is addition of oxygen, removal of hydrogen and removal of electron. Whereas reduction is removal of oxygen, addition of hydrogen and addition of electron, right? For a photosynthetic organism, the source of electron is a chemical species that get excited by absorbing the light rays. Whereas in non-photosynthetic organism, the source of electron is a reduced food, like glucose. Why? Because the chemical formula of glucose is C6H12O6. So it is rich in hydrogen. Hence, glucose is a reduced compound. Clear? This we already studied in the part 1 of biological oxidation and reduction. Now let's come to the today's topic. Oxidation and reduction can be described as half reaction. Now let's take this example where Fe2 plus reacting with Cu2 plus and reversibly gets converted into Fe3 plus Cu plus. Now what is happening here? Here Fe2 plus is getting converted into Fe3 plus. That means Fe2 plus is donating one more electron and hence getting converted into Fe3 plus. Fe2 plus donated one more electron and converted into Fe3 plus. Two reactions are happening here. In the one reaction, Fe2 plus is donating an electron and converted into Fe3 plus. In the second half of the reaction, this donated electron has been taken up by Cu2 plus and hence the Cu2 plus gets converted into Cu plus. So these are the two half reactions. In the first half reaction, Fe2 plus is donating electron. In the second half of the reaction, Cu2 plus is accepting an electron. So who is donating? Fe2 plus is donating. So the substance or the molecule that donate electron are known as reducing agent. Why? Because of Fe2 plus only, Cu2 plus is getting reduced. If Fe2 plus do not donate any electron, then Cu2 plus can't accept electron. So Cu2 plus is accepting electron and getting reduced because Fe2 plus is donating an electron. So Fe2 plus is a reducing agent. Because of Fe2 plus, Cu2 plus is getting reduced. Because Cu2 plus is accepting an electron. Acceptance of electron is reduction. Donation of electron is oxidation. So Fe2 plus donating one electron and oxidized into Fe3 plus, whereas Cu2 plus is accepting an electron and getting reduced into Cu plus. So Fe2 plus is a reducing agent, whereas Cu2 plus is a oxidizing agent. Clear? But if we see the first half of the reaction, in this reaction also, Fe2 plus is a reducing agent because it is donating electron. But what about Fe3 plus? It became oxidized, right? So now it can accept electron because it has space to accept electron also. It can accept electron and again get converted into Fe2 plus because it is a reversible reaction. So Fe2 plus is a reducing agent, whereas Fe3 plus can act as an oxidizing agent. So this makes one conjugate pair of reducing and oxidizing agent. Fe2 plus reversibly converting into Fe3 plus electron. So Fe2 plus and Fe3 plus forms a conjugate pair of reducing and oxidizing agent. Similarly, in the second half of the reaction, Cu2 plus is a oxidizing agent because it is taking electron, and Cu plus is a reducing agent because it can donate electron. So in the second half also, Cu2 plus and Cu plus makes a conjugate pair of oxidizing and reducing agent. Clear? So oxidation and reduction can be described as half reactions. One half constitute of one conjugate pair, second half constitute of second conjugate pair. In the first half, the first conjugate pair is Fe2 plus and Fe3 plus. Fe2 plus can donate electron, Fe3 plus can take electron. So Fe2 plus is a reducing agent, Fe3 plus is an oxidizing agent. Similarly, in the second half of the reaction, the conjugate pair is Cu2 plus and Cu plus. Cu2 plus can take electron, therefore Cu2 plus is an oxidizing agent, and Cu plus can donate electron. So Cu plus is a reducing agent. So here also it makes one conjugate pair. So whenever an oxidation and reduction is happening, it is actually a two-half reaction in which one conjugate pair donate electron and the other conjugate pair accept electron. One of the component of one conjugate pair donate electron and one of the component of another conjugate pair accepts an electron. Now come to biological oxidation and reduction. Biological oxidation often involves dehydrogenation. As we learned now, which compound is known as a reduced compound? Which contains more hydrogen. When hydrogen is added up to any compound, or the compound rich in hydrogen is a reduced compound, the compound rich in oxygen is a oxidized compound. Now, electron can be transferred from one compound to another compound in one of the four forms. It can transfer in the form of electrons only. Electron can be transferred in the form of electron only. Electron can also transfer from one compound to another compound in the form of hydrogen atom. Why? Because hydrogen atom contains one proton and one electron. So if from one compound hydrogen atom is removed and is added to another compound, then in that compound one electron is also getting added up. So that compound will get reduced. Either we can say in the form of hydrogen also, that because of hydrogen the compound get reduced, or we can also say indirectly in the form of electron also, that because hydrogen is added up and because hydrogen contains one proton and one electron, that means in that compound one electron is also added up. So electron can be transferred directly in the form of electron also. Electron can be transferred in the form of hydrogen also. The third form is hydride ion. Hydride ion means H minus. So that means it contains two electrons. Or oxidation can also occur, or electron can also transfer in the form of oxygen also. Clear? So these are the four forms or four different ways by which electrons can be transferred from one compound to another compound. The compound from which the electrons or hydrogen is being removed, that compound becomes oxidized. The compound in which hydrogen or electron is being added up, that compound will get reduced. Reduction potential measures affinity for electrons. We learned that, say for example, the compound 1 is donating an electron to compound 2. That means it can only happen if 2 has more electron affinity compared to 1. 2 can donate electron to compound 3 if 3 has more electron affinity than 2. So the flow of electrons or the transfer of electron from one compound to another depends upon the electron affinity of the acceptor. The acceptor should have more electron affinity, then only it can accept from the donor. And this affinity can be measured using reduction potential in volt. The formula is reduction potential at any concentration is equal to reduction potential at standard concentration plus RT divided by N, that is number of electrons transferred, into F, Faraday constant, into concentration of electron acceptor and concentration of electron donor. So that means reduction potential depends upon the number of electrons transferred and also on the concentration of electron acceptor and donor. At 25 degrees centigrade, as because R is constant, T is constant and F is constant, so in place of R, T and F, we can directly put the value of 0.026 volt. We are talking at biological pH 7 because the pH of our cell is 7. Now, electrons will always flow towards positive reduction potential. Positive reduction potential is as because reduction potential measures the affinity for electrons. That means more positive value means more affinity. If more affinity is there, it can accept electrons from the donor compound. Clear? For example, suppose there are two compounds, compound 1 and compound 2. So to find out in which direction the electron will flow, or to find out whether 1 can donate electron to 2 or 2 has to donate electron to 1, we need to see the reduction potential value. Say for example, if the reduction potential value for 1 is minus 0.192 and the reduction potential value for 2 is minus 0.392. Among these two, which value is more positive? Minus 0.192. The reduction potential minus 0.192 is more positive compared to minus 0.392. Now you tell me in which direction the electron will flow, or among compound 1 and 2, which can act as an electron acceptor? 1. Because electrons will flow towards more positively. More positive E means more affinity for electrons. So the compound 1 has more affinity of electrons compared to compound 2. Hence compound 2 can donate electron to compound 1. So in this case, 1 cannot donate to 2. But 1 can accept electron from 2. So because 1 has more positive reduction potential value compared to 2, therefore the electron will flow or electron will transfer from compound 2 to compound 1, not from compound 1 to compound 2. Clear? So by seeing the reduction potential, we can say in which direction the electron will flow. So if we talk about this chain that 1 is donating to 2, 2 is donating to 3, that means 2 should have more positive reduction potential than 1. 3 should have more reduction potential positive value compared to 2. Okay. Now, standard reduction potential, we can also use it to calculate the free energy change. Now, the formula for this is delta G is equals to minus NF delta E, or if we are talking about standard condition, that delta G dash degree is equals to minus NF delta E dash degree. Now, for example, you have given this reaction, acetaldehyde plus NADH plus H plus giving ethanol plus NAD plus. The condition given is 1 molar each. The concentration of all the solutes are 1 molar. Now, this reaction is again two half reaction. In the one half, what is happening? Acetaldehyde is taking 2H plus and 2 electron and getting converted into ethanol. The reduction potential is mentioned. Similarly, for the second half, NAD plus is taking 2H plus plus 2 electron and getting converted into NADH plus H plus. Or we can say this, here what it is written, acetaldehyde plus NADH. That means what is happening? NADH is giving H to acetaldehyde. So in the first half of the reaction, the acetaldehyde plus 2H plus 2 electron, it is written, this is donated by NADH, and hence NADH is getting converted into NAD plus. So NADH is giving H to acetaldehyde, because of which acetaldehyde is getting reduced to ethanol and NADH is getting oxidized to NAD plus because it donated H to acetaldehyde. Clear? These are the two half of the reaction in which acetaldehyde is getting converted into ethanol. You have already given reduction potential for both the reaction. Now how you will calculate free energy change for that? So first we have to calculate delta E. Right? So delta E is acceptor minus donor. E of acceptor minus E of donor. We learned that electron flows towards more positive reduction potential value. So 0.197 minus 0.197 will act as an acceptor and minus 0.320 will act as a donor. So, and the number of electrons participating here is 2 or transferred here is 2. The value of N will be 2. Now we also know the value of delta E, that is 0.123 volt. So we can put these values in these formula to get the answer. Now what if the concentrations are differ? Here the concentration is 1 molar each. So we have taken the standard concentration. If the concentration of acetaldehyde and NADH are 1 molar, but the concentration of ethanol and NAD plus are 0.1 molar. So the condition is not standard. Then in that situation, how we will calculate delta G value? So first we have to calculate E. So first we have to calculate the reduction potential. Then only we can subtract E of acceptor from E of donor to get the delta E. So first we have to need E for acetaldehyde also, for NADH also. Clear? So the formula is E acetaldehyde is equals to E standard plus RT divided by N Faraday constant, concentration of acetaldehyde divided by concentration of ethanol. This formula we learned in the last slide only. Putting all the values, we'll get the value of E for acetaldehyde. Similarly, for NADH also, putting all the values of NAD plus and NADH, this remains the constant. So we'll get the value of E. After we get the value for E, reduction potential for both acetaldehyde and NADH, then we'll calculate delta E. The formula is E of acceptor minus E of donor. Once we get delta E, we can put in the formula of delta G and can get the result. Clear? Now, cellular oxidation of glucose to carbon dioxide requires specialized electron carriers. As we know, the source of electron in non-photosynthetic organism is a food, glucose, or the reduced compound. When this reduced compound get oxidized to CO2 and H2O, during the process of oxidation, this oxidation is not a single step process. Conversion of glucose to CO2 and H2O requires glycolysis and Krebs cycle. So it's not a single step process. It is an enzymatic reaction consisting of multiple steps. In those steps, some steps are oxidation step, which releases electron. These electrons enter into electron transport chain with the help of some carriers that carry electrons to the electron transport chain or through the electron transport chain. Okay, these carriers are NAD plus, NADH, or NADP plus, NADPH, or FAD, FMN, and ubiquinones. These are specialized electron carriers that carry electron released during the oxidation of a reduced compound or food. Okay. So what are the coenzymes and proteins that are universal electron carrier? NAD plus, NADP plus, FMN, FAD. These are all water-soluble coenzymes and they undergo reversible oxidation and reduction reaction, in which NAD plus and NADP plus, they can move freely from one enzyme to another. They are not bound to any particular enzyme. They can move from one enzyme to another. Whereas FMN and FAD, they are generally tightly bound to the enzyme called flavoproteins. Clear? FAD and FMN remains tightly bound to the enzyme to which it functions, known as flavoproteins, in the form of flavoproteins. Whereas NAD and NADP, they are not bound to any particular enzymes and hence can move from one enzyme to another. Apart from these, ubiquinone and plastoquinone, these are lipid soluble electron carriers that work in non-aqueous environment. That means it present in membrane. Next is iron sulfur proteins and cytochromes. These are also tightly bind prosthetic groups. Clear? So electron carriers are the submolecules that carry electron released during the oxidation of glucose. Oxidation of glucose is a multi-step enzymatic reaction in which few steps are oxidation steps that releases electrons. These released electrons are then carried through electron transport chain with the help of electron carriers. These are NAD plus, NADP FMN, FAD, ubiquinone, plastoquinone, and iron sulfur proteins and cytochromes, in which NAD and NADP plus, they are not bound to any enzyme. They can move from one enzyme to another. FMN and FAD, they remain tightly bound to the enzyme called flavoproteins. Ubiquinones and plastoquinones, these are lipid-soluble electron carriers and hence work in a non-aqueous environment of membrane. Apart from that, two more is iron sulfur protein and cytochromes. They remain tightly bound as prosthetic groups. Now, NADH and NADPH, they act with dehydrogenases as soluble electron carrier. As we learned that biological oxidation mainly involved dehydrogenation, removal of hydrogen, right? So NADH and NADPH, they work with the enzyme called dehydrogenases. So these types of enzymes, dehydrogenases enzymes, what they do, they are removing hydrogen from the substrate. So they help in oxidizing the substrate. Clear? NAD plus and NADP plus, these are called pyridine nucleotide because the nicotinamide ring they contain that resembles pyridine. Hence, they're known as pyridine nucleotide. The source of these electron carriers is vitamin niacin. Now, NAD plus and NADP plus, these two is the oxidized form. NADH, NADPH, these two is the reduced form, right? Because it contains H. So NAD plus accepts one H from the substrate, because here you see 2H, it gained 2H from the substrate, but with NAD only 1H is getting attached. So 1H is released in the aqueous environment. So what these NAD do, they help to oxidize the substrate because they take up hydrogen and electron from the substrate and get reduced to NADH. Similarly, NADP plus, they also can take one hydrogen and one hydrogen released in the aqueous environment. So they also can take hydrogen and electron from the substrate and hence help to oxidize the substrate. These reduced form can absorb light at 340 nm. Now, NAD plus NADH. The concentration of these electron carriers in most cell it is 10 to the power minus 5 molar, whereas the concentration of NADP plus and H together, the concentration is 10 to the power minus 6 molar. Clear? So in a cell, the concentration of NAD plus NADH is more compared to NADP plus NADPH. If you see the ratio between NAD plus and NADH, that means oxidized and the reduced form of NAD, oxidized form is more. Clear? That means it helps to oxidize the substrate. In a cell, the concentration of oxidized form of NAD is more compared to its reduced form. That means it helps to oxidize the substrate because it can take H from the substrate. If we see for NADP, then we can see that the concentration of NADPH is more as compared to NADP plus. That means NADPH mainly helps to reduce the substrate. It transfers hydrogen to the substrate. What the function of NAD? It takes hydrogen from the substrate because the concentration of NAD plus is more than NADH. Whereas in NADP, we can see the concentration of NADPH is more compared to NADP plus. So it can donate H. So it donates H to substrate. So it helps to reduce the substrate. Whereas NAD plus helps to oxidize the substrate. So NAD plus functions in oxidation. NADPH function in reduction. NAD plus, hence it is a part of a catabolic reaction because it oxidizes. NADPH is a part of an anabolic reaction because it reduces. Oxidation occurs in mitochondria. So NAD plus found more in mitochondria. Whereas NADPH, that is anabolic process or reduction, occur in cytosol. For example, fatty acid synthesis. So NADPH is found more in cytosol. Clear? The difference between NAD and NADP. In a cell, if we compare these two, then this is more compared to P form. If we compare only the reduced and oxidized state of NAD and NADH, then we can see that NAD is more in a cell compared to NADH. So that means if this is more, it can take hydrogen from substrate. So if the hydrogen is removed from the substrate will get oxidized. Clear? So it helps to oxidize the substrate. If between the reduced and the oxidized state of NADP, so this is more in a cell. If this is more means it can donate H to substrate. So it helps in reduction of substrate. So it helps in catabolic process, it helps in anabolic process. It is found more in mitochondria, it is found more in cytosol. Clear? These are few examples of various enzymes using form of NAD or NADP. Next is flavin nucleotides, that is FAD and FMN. These flavoproteins, these are enzymes that catalyzes oxidation and reduction using FMN or FAD. These are derived from vitamin, riboflavin. These nucleotides can accept 1 or 2 electrons in the form of hydrogen. These are more versatile, or we can say these are used in more reaction compared to NAD linked dehydrogenases because it can participate in 1 or 2 electron transfer. NADP linked dehydrogenases, they can take 1 hydrogen only, but these compounds can take 2 hydrogen. So these are used in more reactions. If we talk about their absorption maxima, they have absorption maxima at various ranges, like 360 nm in case of fully reduced or 450 nm in case of partial reduced. What do we mean by fully reduced and partial reduced? FADH is partial reduced. FADH2 is fully reduced. Similarly, FMNH partial reduced. FMNH2 is fully reduced. Because it can take up to 2 hydrogen, so it can be in 2 form, either FADH or FADH2. Clear? Now these nucleotides remain tightly bound to a protein. Exception is there, for example, succinate dehydrogenase. In case of succinate dehydrogenase, these FAD, FMN, they are not tightly bound to the enzyme. Because these enzymes are bound tightly, hence they are called prosthetic groups. These are the list of few enzymes that uses FAD or FMN. Now one question from my end for you all. Will the following reaction proceed in the given direction? The direction is malate plus NAD plus giving oxaloacetate plus NADH plus H plus. You have to tell me whether this reaction will proceed in the forward direction or not. How you will get the answer? You have to see the structure of malate. You have to see the structure of oxaloacetate. Then you have to decide whether this NAD plus can take electron or can take hydrogen from malate to get converted into NADH, and after taking hydrogen from malate, is the structure of malate is getting converted into the structure of oxaloacetate. So you have to see the structure of malate as well as the oxaloacetate. Clear? Let me know in the comment section. If you found the video informative, please do like, share and subscribe to the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.